team members. So it's really my pleasure to present and talk something about CRISPR-Cas and how it is impacting crop improvement worldwide and why plant scientists are crazy about this technology. It's a new tools. So I would share all those things, how it is impacting. And then let me begin my presentation with this slide published in Transgenic Research recently. So we have a lot of problem now in front of us, that one of the problem and the base of the problem is the growing population. So we are expected to reach 9.8 billion by 2050. And because of this growing population, we would battle for water and we would battle for the arable land. Also, you know, another very threatening challenge to us is climate change, because the climate change is severely impacting agricultural production by many means. So because increasing average global temperature causes the changes in the dynamics of pest and pathogens. So you can see here, the increase in the amount of global crop production lost to pest is around 10 to 15%. It was at 2016, but it would be around 20 to 25% because of this kind of aggravated climate changes. So we have a lot of things to do in order to feed the growing populations, in order to keep pace with the growing populations. So we have to, the plant breeders and the plant scientists has to improve the plant varieties that could withstand all those problems. So let me share my outline of today's presentation. So in part one, I would briefly mention that agriculture itself is not truly a natural process. So, and the modern crop, what we eat today, where from did they come? And then I would briefly explain in one slide the methods to improve our crop. So what are the methods that we have been using for a long time, and what are the current tools and methods to improve our crops? And in part two, I would go to introduce you to this exciting tools, CRISPR-Cas9, and what is CRISPR-Cas9 and how it is impacting and what can it do for crop improvement. And in part three, I would try to answer, or I would try to justify the title of today's presentation, that is CRISPR-Cas9, how it is rapidly accelerating the crop improvement. And in part four, I would brief about how are genome edited crops regulated. You know, until and unless we address this regulation, then the tools used for crop improvement, and we come up with very improved varieties, but until and unless we can cross the regulation barrier, we could not translate the effort in our lab to the field. Okay, so I would also try to briefly discuss that point. So, you know, what I said, agriculture is not truly a natural process. You'll be understanding from these slides and the next one or two slides. So, you know, the first, according to paleobotanists and all the other scientists, those who were studying the early agriculture, rise of early agriculture and the origin of agriculture, and it was 10,000 to 9,000 BC ago, in the Middle East, that is called Fertile Crescent, they harvested fast agricultural crops in the form of maybe emmer wheat or einkorn wheat or rye. And then from 9,000 to 1,000 BC, this long duration, in this long duration, all around the world, different countries have domesticated different crops. So like in Eastern Asia, we domesticated rice, and other crops have been domesticated in other parts of the world. So this is the duration when many crops have been domesticated. So what is domestication? Domestication is nothing but which would be something suitable for growing in our small agricultural land, and which would be better in taste, which would be better to grow, which would be easy to grow. So this is called crop domestication. I would go into details a bit later. And then comes this great experiment from Gregor Johann Mendel, And from 1856 to 1863, he studied 28,000 peas plant, and he actually performed the experiment on plant hybridization. He published his paper, and later that became the laws of inheritance, like laws of segregation and laws of independent assortment. And he discussed in his paper that one invisible factors is actually uh, playing important role in that inheritance. So what we now recently know as a genes, that has been called by Mendel as an invisible factors. So we'd go detail into genes and later in my slides. So that was one of the breakthrough, how actually when you hybridize two plant and then the next generation comes, and how the trait becomes inherited from one generation to another generation, that became a little bit clear from that experiment. Now, next leapfrog in the crop improvement program is the green revolution that has happened in between 1940s to 1960s. And this is the photo of Norman Borlaug with wheat. So this green revolution is nothing but the development of semi-dwarf high-yielding cultivars for wheat and rice. So, if the plants are very tall, so they are susceptible to lodging. So they bend towards the soil, and then the panicle and the cross become mixed with the soil, and a lot of problem comes during harvesting. So it causes severe losses in the yield. So that problem has been solved by developing the semi-dwarf cultivars in rice and wheat. So, when plant actually uses their energy to grow taller and taller, and if you make them semi-dwarf, then the energy is converted to make more grains instead of making it more taller. So that was the basis of the green revolution, how the semi-dwarf rice or semi-dwarf wheat actually yielded far, far better than the traditional varieties. So now in 1953, in what happened in the green revolution, that was due to the inheritance of a favorable characters, and that increased the yield. But we did not know that it is because of gene and all those other things. But in 1953, after the discovery of DNA double helix by Watson and Crick, this is a DNA double helix model, and this is a photo of Watson. So I met him in Cold Spring Herbal Laboratory. So he was a very nice guy. So after this DNA double helix, we go into details of molecular biology and how DNA and its components actually regulating the expression of gene and ultimately regulating a phenotype. So it's now becoming more and more precise. So if you see this slide, it would be clear to you that all the agricultural produce that we eat today is not due to natural evolution. It's due to the intervention made by plant scientists and farmers, So if you see this, the middle figure is from Brassica oleracea, a common wild mustard. And when farmers or scientists have selected for terminal bud, it gives rise to cabbage. And when they have selected flower cluster, then it gives rise to cauliflower. And when you select for stem and flower, then it gives rise to broccoli. And same like kohlrabi versus sprout and kale, when you select for different parts of that Brassica oleracea, and that actually you desired for that character. So you select for the character from generation after generation, and then you come up with the with this kind of different kind of vegetables that we eat today. So another very remarkable example is the selection for developing today's maize. So if you see, this is the age-old teosinte that has very few numbers of kernel in one cob. So now if you see modern corn, it has huge, even 1,000-fold more number of kernel in one single cob. So this is because of human intervention. It's not that it is happened naturally. So agriculture and agricultural produce that we eat today, most of them are because of human interventions and artificial selection. So now if I want to go in details how those phenotypes are actually are controlled by DNA, controlled by the invisible factors that has been described by Mendel, that we know as a gene now. So if you see any eukaryotic organism like plant, rice, wheat, barley, rye, and even in human body, all the higher eukaryotes, they are made up of trillion of cells. And they are the mass of trillion of cells. So if you see, this is an example of plant cell. And this plant cell has a nucleus, which is one of the main functionary body of this cell. So in that nucleus, chromosome is present in that nucleus, and this chromosome is nothing but is made up of a long DNA thread. So this DNA thread, this long DNA thread is the genetic material of most of the organisms, most of the higher eukaryotes. Now the few part of the DNA are functional and the other parts are not functional according to the current knowledge. So the functional unit of a DNA is called a gene, and that gene actually expresses a character by producing a protein. So this functional unit governs the character in a given organism. So that functional unit is called gene. So if you simply want to define gene is a functional unit of DNA. Now it is clear to us. So what makes the gene and what makes the DNA? If you see in details in the DNA, it is a sequence of 4 different letters that's called nucleotide base. That is adenine, guanine, cytosine, and thymine. So they make the different kind of sequences make genetic code, and that genetic code actually governs different characters in a given organism. So now, if you see the Green Revolution, I have told you, and we have seen that Green Revolution was a conversion from a long varieties to a semi-dwarf varieties, and that yielded more than their taller varieties. So now it was visible through phenotype, and that was done by phenotypic selection. So now later, when molecular biology improved a lot, then we could understand that because this shift from a lodging sensitive to a lodging resistant variety, that is taller to semi-dwarf, was because of a gene that is called semi-dwarf gene SD1. So this lodging sensitive has a fully functional SD1 gene, whether the semi-dwarf one has a truncated SD1 gene. So this loss of function is actually causing the reduction in height of that plant. Similarly, in case of wheat also, in Green Revolution varieties, this RHTB1B and RHTD1B, that is RHT means reduced height gene. So that gene played the important role in developing semi-dwarf wheat varieties like the SD1 in rice. So we are at a place to select our variety or go for artificial selection based on phenotypic characteristics. So now we know what actually causes that phenotype. So here, the 2 classical example is here. One is SD1, another is RHT1 gene in case of wheat. So if you see how crop breeding methods is actually moving towards precision and more precise it is becoming. So if you see, we are solely dependent on crossbreeding, and it takes 8 to 10 years to develop a variety. So if you want to develop a disease-resistant variety, so you have to cross 2 varieties. One is the elite variety that you want to make it resistant, and another is a donor variety. This is the donor variety that has the resistance power. So you have to cross them, and then the problem of this crossbreeding is, it has a lot of linkage drag. What is linkage drag? Suppose you want to introduce one desired character from the donor variety to the recipient variety, but that desired character is governed by a gene. So when you are transferring that character, lot of undesired character may also come together with the desired variety, with the desired character. So it has a lot of linkage drag problems. So you want to bring one important character, suppose submergence tolerance. The classical example of developing a submergence tolerant rice variety. So submergence tolerance comes with brown furrow inhibitor gene, so that the husk color become changed. because it will never be segregated out, the two controlling genes, because they are, submergence locus and the undesired character, they are so tightly linked that you cannot separate them. So, and it is also very labor intensive, this method. So you go to 5 to 7 cycles of backcross to purify the background, but still there is the problem of linkage drag. So you could have the problem of undesired character in your elite variety. So now comes mutation breeding. So mutation breeding is a random procedure of mutating gene or mutating DNA. So you apply to your plant some kind of mutagens, like physical mutagens, like radiation, all the radioactives, and then chemical mutagens like EMAs, ethyl methane sulfonate, all the chemical mutagens, and then you apply them in a whole. It's not at all a precise thing, and then you select for the desired character. Suppose you developed 50,000 population of a mutant population that has been developed from radiation, mutagenesis. And now you have to screen for phenotype, what kind of phenotype you are looking for. Then you select that plant, and then you go for repeated backcrossing to purify that one, and then you develop a variety. So, but this has a problem, same. Screening is expensive and then labor intensive, and the mutation is random. You don't know in what other genes have been also modified due to that radiation, along with your desired character modifying mutation. Okay, so this is a gamma garden that has been in use after World War II. So this the central part is the source of radiation, and those are the plants that have been that are intended to treat with the radiation. And then you take out those plants and collect seed and grow next generation, and then you see a lot of variation because of the several kind of damages happen in their DNA, and then you select for the desired one. So this is mutation breeding. This is so random, but these two are not regulated. Okay, so next comes the transgenic breeding, or that we all popularly know as a GMOs, genetically modified organism. So it has unique advantage over the other two earlier methods. The advantage is that you could use the whole gene pool. You are not restricted to the gene pool of the same species. Like, here, if you want to hybridize two wheat plant, so hybridization and mutation breeding can generate variation from the wheat gene pool only. So you cannot hybridize a rice with wheat, or a mango with banana, okay? So it cannot be done. But in case of transgenic breeding, you could use the whole gene pool because even you could use a mango gene to introduce into your rice genome for a desired characteristic features that you want to develop. Now, but it has its own problem. The problem is the integration loci is uncontrollable. So if you, when you insert foreign DNA or foreign gene into your variety of interest, but where that gene is getting integrated, in which part of the genome, is not controllable. You cannot control that, okay? And it has because of lot of fear mongering, things from the opponents, and non-science based argumentation that actually causes developing the very stringent regulatory scenario in the whole world. So it has a problem of this regulatory red tape, and also it is a high cost is involved to develop varieties using the transgenic technology. Now comes the genome editing technology, and mainly CRISPR-Cas. And this CRISPR-Cas actually can give rise to a predictable and precise variation that you want to want to develop. And it has the high potential to escape GM regulation because in many countries it has already been started. They started to deregulate certain kind of genome edited crops. And you could develop a variety using CRISPR-Cas9, which would be foreign DNA free. So you don't have to have a foreign DNA inserted into the genome. So no need to keep that foreign DNA or foreign construct into the genome. So it would be a foreign DNA free one. So it gives us a lot of hope. So I would be mainly discussing this CRISPR-Cas in my later part of presentation. So now coming to part 2, and now I would introduce you to this exciting tool, CRISPR-Cas9.